Hello, everybody. We are now understanding a question from the GATE Biotechnology 2020 paper. It is related to the topic recombinant DNA technology. So, the question is which one of the following statements about Agrobacterium ti plasmid is correct? Option A, Vir genes are located within the tDNA segment. Option B, phytohormone biosynthesis genes are located outside the tDNA segment. Option C, opine catabolism genes are located within the tDNA segment. And option D, opine biosynthesis genes are located within the tDNA segment. So, before we answer this question, to understand what agrobacterium is, let us first understand that gene transfer in plants can be done using two common ways. So, one is agrobacterium mediated gene transfer, where a gene of interest is carried by the bacterium which delivers it into the host cells. And another method is by bombarding the cells with particles coated with the gene of interest. Now we'll look more into detail about the first agrobacterium mediated gene transfer. So, Agrobacterium cumifaciens is a bacterium known for its ability to transfer a part of its DNA, known as tDNA, into plant cells, leading to the formation of plant tumors called crown galls. So, this natural mechanism has been harnessed for genetic engineering purposes to introduce desired genes into plant genomes. So here are some practical examples of genetic engineering using Agrobacterium tumefaciens to help engineer crops with desirable traits like herbicide and pest resistance, vitamin and nutrient enrichment. That is, they have been used to introduce genes responsible for the synthesis of specific vitamins or nutrients into the plant species that may lack them. For example, golden rice was developed to produce provitamin A, that is beta carotene, thereby addressing vitamin A deficiency in regions where rice is a staple food, given the fact that rice does not have vitamin A. And then to provide disease resistance for improved crop quality and yield. Like, for example, they've also been used to modify the quality of the crops. For instance, researchers have developed tomatoes with delayed ripening to extend shelf life and potatoes with reduced levels of certain scratches for improved processing. So, moreover, they have also been used to deal with environmental stress tolerance, bioremediation, and phytoremediation, and for synthetic biology purposes. So, this is the structure of a TI plasmid. All the TI plasmids code for functions associated with plasmid replication and maintenance, conjugative transfer, virulence, opine utilization, and sensory perception of exogenous signals released by the plant host and neighboring agrobacterium cells at the site of infection. So, now let us look into the various parts, the components of the TI plasmid, and look at their roles. So, the first one is the Vir region or the virulence region. So, the Vir region of the TI plasmid encodes a set of virulence genes that are responsible for several important steps in the infection process. These genes are involved in the production of proteins that form a type 4 secretion system which transports the tDNA and other virulence factors into the plant cells. Then we have the tDNA, which is the abbreviation for transfer DNA. The tDNA is the region of the TI plasmid that is transferred from agrobacterium into the plant cell. It contains genes that manipulate the plant's cellular machinery to promote growth of tumors and to create an environment suitable for the bacterium's survival. The specific genes carried by the tDNA can vary, but they often include genes for biosynthesis of plant hormones like auxins and cytokinins, and opines, which are amino acid derivative that serve as nutrients for the bacterium. So many TI plasmids carry genes responsible for the synthesis of opines, which is inside the tDNA region, and for the catabolism of opines, which are responsible for breaking down and utilizing these opines. So, opines are basically unique compounds that are produced by infected plant cells and serve as carbon and nitrogen source for the bacterium. The opine utilization genes enable the bacterium to use opines as nutrients, giving it a selective advantage. So, apart from these, we also have the origin of replication, the ORI. The origin of replication region of the TI plasmid serves as the starting point for DNA replication. It is necessary for the plasmid to replicate itself as the bacteria multiplies. And then the TI plasmid also has promoters and regulatory elements. So these elements, they control the expression of genes in the Vir region and the tDNA. These elements ensure that the genes are expressed at the appropriate times during the infection process. So some TI plasmids also contain selectable marker genes, often encoding antibiotic resistance. These markers are used to select and identify bacteria that have successfully acquired the plasmid. Also, moreover, whatever the gene of interest is, it is inserted into the tDNA region so that it gets transferred into the host. Now, coming back to our question. So, which one of the following statements about Agrobacterium TI plasmid is correct? So, option A, Vir genes are located within the tDNA segment, which is false. It's not correct because virulence region is a separate region apart, like outside the tDNA region. So we rule this out. And option B is phytohormone biosynthesis genes are located outside the tDNA segments, which is again false. So yeah, we have the phytohormone biosynthesis genes that produce auxin and cytokinins within the tDNA region. So moving on to option C, which is opine catabolism genes are located within the tDNA segment. This is again false because we just saw that the opine catabolism genes are located outside the tDNA segment. So now we are left with option D, which is opine biosynthesis genes are located within the tDNA segment. So yes, this is the correct answer. We saw that the genes responsible for opine biosynthesis genes are located within the tDNA segment. So the correct answer for this question is option D. Thank you.